Hello, everyone. Welcome to our monthly Science of Alt Protein seminar. My name is Amy Huang, and I'm the Associate Director of Scientific Ecosystems at the Good Food Institute. For those of you who are here for the very first time, welcome to our community. GFI is a nonprofit think tank that is 100% driven by philanthropy, and our goal is to build a world where alternative proteins are no longer alternative. If you're looking to take a deeper dive into the science of plant-based fermentation or cultivated meat technologies, check out our open access resources at gfi.org/science. We are launching new publications and tools and resources all the time to help support you in your work. So with that, I will move into introducing our phenomenal speaker today, Dr. Rosenvald. Dr. Rosenvald is a highly experienced scientist and executive in the alternative protein space. She has a PhD in food technologies, specializing in the molecules behind the taste and aroma of food. For the last 10 years, she's been running multiple research projects and leading research teams in TFTAK. In recent years, she has been focused on protein research and meat alternative developments, helping to develop and commercialize multiple alternative protein products. Her team has been granted 2 GFI research grants and is currently participating as a finalist in the X Prize Feed the Next Billion competition. Sirli is also the co-founder and CEO of FUNKI. FUNKI provides taste and healthy high-protein food products from mycoprotein to diversify people's everyday funky choices, or everyday food choices rather, and nudge their consumption habits towards more sustainable paths. As a flavor scientist, she is passionate about bringing to the market sustainable products that people love for their good taste, and we are thrilled to have her join us today to run through how we can really help plant-based protein in the form of salmon fillets achieve sensory parity with its animal origin counterparts. So with that, I will turn the floor over to Dr. Rosenvald. Thank you again so much for joining us today.
Thank you, Amy, for a very nice introduction. Before I move to the topic, so Amy introduced me, but a few words about TFTAK. So TFTAK is a contract research organization located in Tallinn in Estonia, and TFTAK is working to accelerate food and biotech innovation. And for recent 5+ years, we have been working with multiple alternative protein technologies, with plant-based, but also different fermentation-enabled technologies, other biomass or precision fermented products. And I'm moving now to the topic that I wanted to discuss with you today is how to really improve fish alternative products. And we are working with all of the aspects of a good alternative product. So starting from aroma, taste, but also how to get the right texture, extrusion technologies, and how really to formulate and make it all to look like a real salmon fillet. So, but today's presentation, my focus will be solely on the aroma part. And although in our everyday work, we actually, especially when working with plants, in case of aroma, from one side, we have to work with flavors, looking at the ways to mitigate, how to mask them. This is one part of the aroma formation, but also how to get true fish-like aroma. And I know that a lot of companies are also struggling how to really get these top notes in their products that could be feasible and will stay as good as original all the way throughout the processing as well. So around 3 years, we have actually working with salmon alternative. And when we look at the composition of Atlantic salmon on the macromolecular level, so it is basically no carbohydrates, but a lot of protein and a lot of fats. So actually the fat content is very, very much fluctuating. So here I have brought out that 10 to 15 grams, but it can actually be lower and it can be also higher. But those macronutrients are also very important in the aroma formation, and especially fats, which is one of the crucial a component where the aroma of salmon and actually all fish is coming from. But if we are talking about high fat content fish, then the importance of fat is of course more and even more important. So when we want to imitate a salmon fillet, where we started was, of course, carrying out GCMS analysis to really bring down the aroma of salmon into molecular level. And we have actually analyzed a lot of different salmon fillets from different seasons, either frozen or chilled. And we have seen salmon fillets with different fat contents. And I can say that the aroma profile of these products are also varying quite a lot. So the salmons, which have very high fat content, they also have much more intensive and aroma profile as well. And when we started with developing fish alternatives, we of course took the original salmon, and we looked our developed products and wanted to understand what is the difference between them and where we could improve. But when we talk about plants, then of course, if in case of salmon, most of the aldehydes, most of these molecules are coming from the fats. So if we talk about plant-based salmon, then these molecules are already present in the basic ingredients. So protein isolates, protein concentrates. So it's very rich in different aldehydes and ketones as well, which are also originated from fat, but from the pea fat. And when we analyzed the raw salmon and cooked salmon with our developed prototypes, both raw and cooked, I'm not going into molecular level yet, but what we noticed was that in case of real salmon, most of the aroma molecules are generated throughout the cooking process. And in case of our alternatives, we saw that the aroma profile was quite similar in the, let's say, raw product as well as cooked product. So the processes are taking place in very, very different way. And this led us to identify our objectives, and we wanted to really see what are the key molecules, because from the GCMS profile you get more than 100 different volatile molecules. So we wanted to really understand which are the important ones in terms of sensory perception, which actually make the salmon taste as salmon. And we already knew that most of the molecules are fat origin, but we went to look into deeper in that as well. And to really be sure what are the key molecules, our aim is still to carry out recombination study, meaning that we will put salmon aroma together, put it together in a lab from pure aroma molecules. And another objective was to understand the formation pathways. So if we know which are the most critical molecules, we want to understand how they are formed in a cooked salmon, and then find possible ways how we could really recreate the same pathways with using different plant-based sources. So once more, that we saw that in case of salmon, the aroma, most of the aroma is created during the cooking process. So fresh salmon is quite neutral and mild in its aroma, but plant-based products hardly change. So we wanted actually to change it. We wanted to try to develop also product that gets its characteristics during the cooking process. Then we moved forward to GC olfactometry. For those who don't know what it is, so basically gas chromatography is separating all the molecules, and human assessors are sniffing them one by one. So this methodology is helping us to understand which of the molecules coming out from the, which of the molecules that are separated really have odor activity, meaning that they really possess odor, and they are therefore important and influencing the sensory properties of a salmon. And this is quite a long list of molecules that we determined with GC olfactometry that are as the key molecules in salmon. This was the preliminary list. So we are now in a process to eliminating molecules one by one. But what we can see from here is that there are a lot of different aldehydes in the salmon, ketones, alcohols, all are mainly fat generated. And what was a bit surprising for us was that you can see only one pyrazine was measured in the, this is actually a result from multiple olfactometric analysis. So this is a combined result from multiple samples. But what we saw was that there is not too big role of Maillard reaction in cooked salmon aroma. So mostly fat-related components, and only one pyrazine, and also meaning that salmon actually has quite low roasted and that kind of aroma notes. And where we used this olfactometric data more was that we used all these pure molecules to train our sensory panel as well, so they could know what are the different characteristics that you can find from salmon. And I will show you in later slides also how our sensory panel has discriminated or has taken into pieces the aroma profile of salmon. So it's not only fish or fishy. But here I have marked some of the molecules with red, and these are actually the molecules that salmon, in order to be like salmon, and in any fish in general, you should definitely have some fishy notes as well. And in most of the fish products, 3-methylamine is one of the molecules that is very distinct, fishy, spoiled fish-like notes. But we have also found that there are two other aldehydes that also very much contribute to the same fishy characteristic. So regarding 3-methylamine, those working with fish alternatives most probably know that this 3-methylamine is one very critical molecule, and we all also knew it already for quite a long time, but our aim was also to understand if there is a possibility to have a fish salmon product without using 3-methylamine. Is this really something that is definitely needed in a product for it to resemble fish? And what we have also seen in this analysis is that in raw salmon, very often this 3-methylamine is missing, but it is always occurring in the cooked salmon. And the thing with 3-methylamine is that it is very common in different fish species and also in algae. And it has a very important role as an osmolyte in this species. And 3-methylamine is reduced from dimethylamine oxide during postmortem changes. It can be through enzymatic pathways, either endogenous or due to the bacterial spoilage. So basically, the fish starts to stink more fishy and fishy during the spoilage. But it is also, according to literature as well, and also according to our experiments, we can see that it is also formed with heating. So during the cooking process, also 3-methylamine concentration is growing. So 3-methylamine is definitely something that is on top of everybody's head who is working with different fish alternative products. But our aim is and was and is to look a bit further from 3-methylamine to really see what are the other important molecules, and what does the aroma of salmon actually mean? So what does it contain? And here is an example of one specific salmon fillet that we have tested. There are, of course, pictures which are a bit different. So as I said, for example, fat content is really crucial, and it is determining a lot what kind of notes you can find in a salmon fillet. But what we can see from here is that the fishy note is really the most dominant one. And for our assessors, if we say fishy, so this is basically trimethylamine. So this is a really fish-like, rotten fish-like distinctive odor, because we have here also marine, which is a bit different. So this is more like reminding algae, sea in general, a bit different type of characteristic. But besides these fishy and marine notes, there is also some roasted notes depending on the product. It is milder, but there are some roasted. Of course, it depends also on the way how it is cooked. Then there is also some green notes, because salmon contains also some green-like aldehydes like hexanal and some other. These are rather low, but again, depending on the sample, it might fluctuate and a bit more, can be a bit more important as well. We have seen that sourness is not very common to salmon, so it's not a very often used descriptor in salmon products. There is some sweetness. There is also some mushroom-like notes or earthy notes, and this again it might fluctuate a bit, but it's more in the lower intensity level. And metallic, which I have actually seen that can fluctuate quite a lot, so some samples can be more metallic. And one very interesting thing as well is the buttery notes, and these are some specific ketones that we are also now, when we are trying to recreate the salmon aroma, we also pay a lot of attention to this buttery notes as well. And this buttery notes, we have also seen some samples where they have actually been quite extensive. And potato, which is also coming from a very, very specific molecule that we have identified. In most of the cases in real fish, it is not very distinctive, and it is more on the lower intensity level. But as you can see, there are different descriptors. So if you, although trimethylamine is a very important molecule, it gives the fishy note, but if you have only trimethylamine in the product, then you are missing all the complexity of the salmon flavor. And trimethylamine plays a more important role on some white fishes, but in case of salmon, the aroma is sometimes actually very, very complex when you start to look all sort of that different aroma nuances that actually are present in there. But when we had seen that the aroma of salmon is mainly coming from its fat part, so fatty acids play a very huge role, then we started to look what could be the possible sources where we could have these same aroma molecules being generated during the process. And we selected linseed oil and hemp seed oil as two plant-based sources, which contain also a number of different polyunsaturated fatty acids. Not the exact same ones, because EPA and DHA, which are very important in salmon, they are missing in linseed and hemp seed oil. But the aroma formation pathways for these other PUFAs is rather similar. So our expectation was that we can generate similar molecules also from the other polyunsaturated fatty acids. And of course, algal oil are very much used in alternative seafood developments. And we have also actually done quite a lot of work with different algae. We have worked with algal oils, and we have also tested different algae and extracted oil out from it by ourselves. And why we started to think about this hemp seed and linseed oil was actually that some years ago, we extruded hemp protein, and quite unintentionally, the product ended up being fishy. And so we started to think that most probably the fatty acid profile in the hemp seed is something that causes this fishy notes. So as we had this experience, we decided why not to invest into it further. But I have to say that we haven't been too successful in that, to use these oils to generate fishy notes, and it depends a lot on the specific ingredient and specific provider, how it actually behaves during processing. We carry out some of the experiments with different oils, and what we basically did was that we extracted aroma molecules with from the headspace with very mild conditions, and we also heated the oil samples for a couple of hours and extracted the volatiles then. And what we saw, of course, for the salmon oil, so this extracted salmon oil, this was oil that we extracted from raw salmon ourselves. So we saw that during the heat treatment, the content of aldehydes and ketones was growing quite a lot. We didn't see almost no change in sunflower and rapeseed oil, which was expectable, but we wanted to still include them. And we see quite a large change also for the hemp seed oils, but surprisingly not for the linseed oil. So the contents actually didn't change very much. And algal oils gave also positive result in a way that the aldehydes that are of our interest, their content was increased quite a lot. But we also did very quick sensory testing also from these samples that we were analyzing, and we were hoping that we can generate some fish-like aroma. We of course measured the molecules, but we also wanted to see if it actually is applicable and if you can really imitate fish. And the only fish-like aromas we got with salmon, of course, and algal oils. So another experiment what we did was, again, for the same reason. We use different pure fatty acids. We use different pure standards, and we also wanted to see what are the molecules that are generated during the heat processing. And again, we wanted to see if some of these samples would possess fish-like aroma characteristics, so we could know that, try to identify the molecules that are most important in there. And we saw quite many differences on the processing profiles of different pure fatty acids, in alcohols, in different aldehydes. So I have selected the molecules here, which we see as the key molecules in salmon as well, and which are of our interest therefore. And we saw different volatiles created, but as we really wanted to see also some fish-like notes occurring, I have to say that this was something that we didn't see. So in that sense, this experiment was not too successful, and we couldn't get any very good applicable result to move further with our experiments. And as I said, from this plant or non-animal based oils, algal oils are the most promising ones. And we saw an article which was actually mapping different microalgae for flavoring in different plant-based seafood. And so different microalgae for screened mainly for all sort of aldehydes and aroma molecules, but we were mainly interested in trimethylamine content. And there was concluded that there were 3 microalgae this Biodactylum trichonutum, Rhodomonas salina, ja Tetraselmis chui, which contain 3-methylamine. And we decided to move forward with that, and we also ordered samples of different microalgae to our lab, and these were the algal biomasses. So we again extracted the oils out of it and carried out again GCMS analysis. We also looked into different interested aldehyde profile, also the ketones. So I'm showing also the ketones because those two, this butanedione and pentadione, these are the ones which are giving this buttery notes, and they tend to be also quite important in the overall salmon profile. But our main interest, of course, was in 3-methylamine content, and we had 2 of the algae samples, which I just showed in the previous article, and we also found 3-methylamine in these samples. The only thing is that these microalgae are green, and even the extracted oil is very green, so it's quite difficult to really use it in alternative seafood applications. And where we moved forward from there is that based on the olfactiometry data that I showed, where we identified the key molecules, what we did next was that we developed a GCMS quantification methodology to really quantify these key molecules in salmon samples. Here are the results of 2 fillets that were quantified against these molecules that we had been identified previously as the key molecules. And based on this data, we created a aroma model, so using these pure molecules and really to put together a salmon aroma from pure chemical molecules. So this is the part of the recombination studies. And the idea here is that we will put together, at first, we want to get a very authentic salmon aroma from these pure molecules. Once we have achieved it, we will start to leave out molecules one by one to determine which ones we can leave out without having major influence on the aroma of the salmon. So this is the methodology, how to really understand which molecules are the most important ones and really necessary. And for this recombination studies, we are using our in-house trained panel, and we have done quite a lot of different fish trainings with salmon, but also with different pure molecules, different spiked samples. But this recombination part has been also with its own challenges. So there has been many decision points that we have had to make. So first of all, how do we sniff the single molecules? There's one option is to use sniffing stripes, but we decided to use sniffing glasses. And at first, we actually dissolve these molecules in water. So all the molecules were put into water and then into the sniffing glass. And what we saw was that if I say honestly, the aroma that we get was not resembling fish too much. And at one point, we got an idea that we could use oil as a solvent instead. And this actually gave us much better results. So when we use the oil as a base to put our key molecules inside, we get much better proximity to the real salmon. And we can see from here, so the OBS is the oil sample and WBS is the water sample. So the recombined fish was much higher in intensity. This is something we had to work also a lot to get it into intensity that it is sniffable in a sniffing class. But what was good was that we really got in the oil-based sample, we got the fishy notes quite close. And also in the water-based samples, we had distinctively high green notes. And in oil-based sample, it was much lower. And also marine was better. And in general, we get much closer profile to fish when we use the exact same amount of each molecule, but in oil instead of water. And we are actually in a middle of this recombination study. But one more thing that I wanted to show you now is that we also, as I said, one of the things that we would also like to get an answer is that if 3-methylamine is something that is really unavoidable and it is a must in all alternative seafood products, which basically means that you have to find either an algal source or you have to use some flavoring agent to have this 3-methylamine in your product. And this was one of the recent tests we made. And there I have, in comparison, the real salmon, the oil-based sample with 3-methylamine, oil-based sample without 3-methylamine, and water-based sample. This is also with 3-methylamine, and we really saw that that 3-methylamine in oil sample with 3-methyl, it gives the best result. In water, for some reason, it is much less fishy. And if we leave 3-methylamine out, so our current recombinant or recombinated salmon is not fishy at all. And this is the same with the green notes. And well, if we look with the marine like, then also the 3-methylamine is the one giving more marine notes in general as well. And now moving forward, I talk a bit what are our plans for the future in this respect. So we are still working in recombination studies. According to assessor feedback, as you saw, there is quite a lot of improvement still. We have too high green-like notes. So although the concentrations were all selected based on the GCMS quantification data, we are now looking into ways to make changes to our recombinant flavor based on the assessor's feedback. So we are starting to lower some specific green molecules and increase some specific aldehydes that also give fish and marine-like notes that I showed you on the table before. And our aim is to really get our recombinated salmon aroma as close to the real salmon as possible, and then we can really start to eliminate some molecules. And when we have got the new concentrations based on the sensory results or sensory determined concentrations for this recombinated salmon, then we can go back to the fatty acid data and really start to look what were the what are the fatty acids that can really help us to enhance the fishy notes. So one of the things that just quite recently got my attention was that the commercial algal oils, at least the samples that we have been worked with, and I think there is 3 or 4 of them, they do have a distinct fish-like aroma, and it's maybe it's lower in a headspace, but if you taste it in your mouth, you can really feel the fishy-like aroma molecules. And they are not, any of them is containing 3-methylamine. So that was one of the ideas that we have done the first preliminary GCO trials to really find that what are the molecules inside these algal oils which are really giving that fish-like aroma. And then we can combine the data with the salmon results. And I'm really looking forward to see where it takes us. And the next thing is that how to find the feasible way to incorporate trimethylamine, because we talk about flavorings, and even when we do these recombination studies, we are seeing that TMA is really, really volatile. So very often, if we do recombination study, in the first sniffs, everybody feels this fishy aroma, but it is flying away really, really fast. So during the processing, it disappears. The same is for our fish alternative sample. We are using an ingredient containing TMA, but when we analyze our fish alternative product after cooking, there is not any left anymore. So it is, although TMA seems like a thing to use, but there are some difficulties related to using TMA on product development process. And when we have finalized our recombination studies and we really get a recombined salmon aroma in our lab, we hope to publish in the end of this year our results on the quantification and the molecules that we have identified as the most important in providing salmon aroma. And our aim is to bring it down to as few as possible. So we are now working around 25 molecules, but hopefully we can bring this number down, and then we can really focus on the specific molecules that are the most crucial ones. So that's all by me for now, and I wish to thank GFI for the grant and to financing this research. And it has been a very interesting process, although not all the result hasn't been so successful as we have been hoping for. But I wish to thank also the whole team in TFTAK who has done and is doing an excellent job. And we are always thinking how to find new ways that we can really find something applicable, and we can really make a change in alternative seafood development industry. So we have a year more to go. So I really hope that we will have something interesting to share after a year. So thank you.
Thank you so much, Sirli. That was fantastic. And we very much look forward to that publication. We are now going to be moving into the Q&A section of today's webinar. For those of you who haven't yet gotten the chance to do so, feel free to drop any questions that you have for Dr. Rosenvald in the Q&A section of your Zoom menu, and we will try and get around to them in the next 15 minutes or so. Alrighty. The first question that we have here is around how did you choose the descriptors for the sensorial analysis?
So basically, it is a combination of two things. The third thing is that we have a trained sensory panel who has quite extensive experience. And from one side, we described everything that we can feel. And the other side is that we did olfactometry analysis. And based on that, we already knew we grouped the molecules into categories. We knew that there are like green fatty notes. We saw the buttery notes, and this is something that was very well coming out from the sensory analysis as well. And so as we knew already the molecules, we had kind of a clue what we could expect for. And then we validated it with the trained panel. And as we quite often train our panel with pure standards, once they learn the descriptions with pure standards, they are much better capable to recognize it in a real food matrix as well.
Thank you. Our next question, what aroma extraction technique did you use to extract the volatile compounds in salmon?
Yeah, so we are using SPME, so solid phase micro extraction from the headspace.
Thanks. There are a series of additional questions around how the sensory profile or evaluation was carried out. I'll ask them piecemeal so that I don't overwhelm you with 6 questions at once, although they are all part of the same. The first is just asking about the preparation of the salmon for the sensory evaluation. Was it cooked salmon? Was it warmly served? Was there salt used? If you can just specify a little bit more how that salmon was prepared.
We have actually used the different preparations. So we started using oven cooking. Why we use oven is that it is much more homogeneous compared to pan frying. But in some of the cases, we are actually oven cooking and then we are homogenizing. And this we do also for the GCMS analysis. So we really know that everybody is getting the same thing, because if you think about the piece from the head part or from the tail part, it varies in fat content. So we are trying to cook it and then homogenizing it to really have a similar that everybody has a similar sample. And as we are not interested in texture or appearance, so we have done like muffins also in the oven as well, homogenized the raw salmon and then cooked as a muffin in an oven. So for different testings, we have actually tried quite a lot of different setups and preparations.
Interesting, thank you. Also, a follow-up question about the sensory profile analysis. Did you all only incorporate kind of olfactory data like smelling, or did you also combine that with tasting evaluations?
So these evaluations are all only olfactory, so we are only smelling.
Great, thank you. There is a question here around the ultimate kind of green color of your final fish prototype. Even though the fish flavor might be on point, do you have any suggestions around what it would look like to remove the off colors of those prototypes into the future?
Others.
I'm sorry, what was that?
So yeah, the question was about off odors.
Oh, it was about off colors. Sorry. So green, this participant is remarking on the green color of the final fish prototype.
Okay, so I wouldn't say that. I would say that the color is a bit more critical in case of whitefish, but in case of salmon fillet, I guess we have managed with careful ingredient selection and by using different colorants, so the color is not an issue for us, at least.
Thank you. There's a question here around what your strategy is to remove the off-flavor compounds that were detected.
I would also say that the first strategy would be to select ingredients wisely, because we are working a lot with legumes and pea protein. And if you look at the commercial proteins, then I would say that today there is 20 plus something pea isolates available, and they all are very, very different. So we have selected smartly our ingredient that we use, and we are of course using masking technologies as well. And I would say that we feel that we are quite successful with that.
Great, thank you. There's one question here around butter aromas. Do you think the use of butter aromas would help to give an authentic salmon taste?
I believe that they are part of that. So the salmon aroma really is a complex. So it's not about getting fish, like I was saying, the dreamethylamine. And buttery notes seem to be also quite important part of it.
Great, thank you. This next question is around whether you might be planning on testing some amino acids like glutamate or aspartate in mimicking the umami flavor or taste of salmon.
Yeah, regarding taste, yes, and this is not only about amino acids, but fish salmon contains also nucleotides. So these are also something that are important part if we talk about salmon experience in general and not only about aroma. But for some of the products, for meat products, for example, for aroma generation, also amino acids are very important. But in salmon aroma, that, as I also mentioned, that we were a bit surprised that the aroma is so much fat dominated. So all the other things are minority.
There's another question here by a fellow GFI grantee. Are the sensory training standards diluted in solvent, or do you use the pure standards? And if solvent is used, what solvent do you use?
Yes, so we are using solvents and we are using ethanol and water, and depends a bit on the molecule. We want to solve more in water, but in some of the cases we need to use ethanol as well.
Great, thank you. A lot of questions here around trimethylamine and maybe the associated literature. So one question is whether there's any literature indication that you might actually find a fishy flavor compound that's different from trimethylamine and that is less volatile than it. And would you expect those compounds to always contain nitrogen?
Yeah, that's a good question. This is a question that I'm looking answer myself as well. I have to say that we have actually also screened a lot of commercial flavorings as well, because we just wanted to see get an understanding if 3-methylamine is like critical and you can't have a fish-like thing without it. But as said, I'm very optimistic due to the algal oils that we have tested, and they are really fish-like. So I hope that we will find something that could be as good as 3-methylamine.
Thanks, Sirli. There's another question here around whether you think it's possible to generate or otherwise produce the trimethylamine in plant-based salmon without actually having to add it.
Yeah, of course, this is something that we have also thought about a lot, and we have looked different pathways, because it is something that is also produced in human organisms in some conditions, and there are some, there might be, but the honest answer is right now that we don't have a clear idea yet if that would be possible. We are kind of looking into it more, I guess, but I can't say if I'm not sure yet if that would be possible or not, but we are definitely looking into that as well.
We look forward to figuring out what we collectively explore or discover. How do you turn, or maybe can you speak to the process of how you turn aromas from microalgae into aromas concentrated in oil?
In the microalgae, we are using just fat extraction process, and a lot of aromas are in the fat. So they come during the extraction process, they stay with the fat. So we are not doing anything specific. It's basically fat extraction process.
Great, that's helpful. Thank you. A couple last questions here. One is about your volatile extraction techniques. Are you using cold aroma extraction, or are you thermally heating the sample for a more volatile extraction?
So we are warming the samples. We have done something in 30 degrees as well, but standard is 40 degrees. Yeah, so we are heating a bit as well.
Great. And then lastly, as you're considering different kinds of compounds for the flavor profile, are there other groups of compounds other than fats than you're exploring and that you were exploring? And if so, what are they?
In case of salmon, I would say that these are olefats and trimethylamine, so they are the core of salmon. But in general, we are working with meat alternatives as well. So we have looked a lot in Maillard reaction between amino acid, sugars, so aroma creation in different food products. So there are different pathways. But if the question was about salmon, then I think the secret is all in trimethylamine and fat. So there is not much more to look into.
Great. That's all for our participant questions. I would love to wrap with one last question for you, which is just, we've got this captive community of alternative protein scientists. What kind of last words would you like to leave this community with as they embark on trying to advance alternative protein science and engineering?
The pricing question. So what I would like to say, I would say that for me it is a very interesting world and a lot of challenges, and I think there has already been a lot of solutions. So I'm really optimistic that we can only work forward together and move forward, and I'm sure that we will have a very good tasting salmon fillet and every other animal protein product available in the markets in the coming years. So I think the future looks bright. There are so many bright minds working on these challenges, and it has to succeed.
Yes, I feel that kind of impatient optimism as well. Thank you so much, Dr. Sirli Rosenvald, for this seminar. Thank you for sharing your research and your findings with this community. It's so deeply appreciated, and we look forward to continuing to follow your work. Please do, when that publication comes out, send it along and we will disseminate it across the community. And thank you to everyone who attended our seminar today. We hope to see you again next month for our monthly Science of Alt Protein seminar next month with Dr. Lutz Grossman. Thanks so much, everybody.
Thank you.